Time Matters, Why PKPD is Important for Oligo Development with Dr. Carl Amilon from AstraZeneca. Thank you very much for agreeing to do this presentation for us, and over to you.
Thank you very much, and good morning, good afternoon, everyone. It's a pleasure to be here, and thank you to the organizers for inviting me to come here and give this talk. It's something that is very close to heart to me, and it's very much appreciated I get this opportunity. So my name is Carl Amilon. I work as a DMPK project lead for AstraZeneca based in Sweden. I'm aligned to our cardiovascular, renal and metabolism disease area. I have a background in clinical pharmacology and pharmacometrics, and nowadays I work within early preclinical development, supporting our portfolio of projects from really about 2 years before we start our first time in man planning until we reach the market with our products. If you have any questions or anything, you can type them in the chat box, and I will try and answer them as we go along. I think it's better if we take the questions as they go along, as they come up, rather than saving them all to the end. If you have thought about something, or if something is unclear, it is probably the same for someone else as well. So then I think it's better that we take them as we go along. So why do we need to know about PKPD, and why is it important to oligo development? I've tried to divide this into 3 areas of understanding, or 3 areas of interest, and it's the ADME part, so it's the absorption, distribution, metabolism and excretion. It is the DMPK part, which is the drug metabolism and pharmacokinetics. Into that I also tie the pharmacodynamics part, which is the effects, and it is for safety, to understand that we have something that is safe and tolerable with sufficient margin at the effective exposure that we are striving for. And the reason why we need this information is to understand when to dose, how much to dose, and who should receive the dose that we are giving. We start this presentation with a crash course in pharmacokinetics and pharmacodynamics, and then we will move over more to the application part, and where we have some common pitfalls, and where I, in my experience, have seen that there is perhaps sometimes a discrepancy between different disciplines on how we look at data, and this is my perspective on how one can think about it. So we define pharmacokinetics as the study of what the body does to the drug. So it is a study of the processes that impact a drug that will generate, of course, an exposure in the body. General for any drug molecule is that we administer it typically orally. It can be subcutaneously or intravenously for other than small molecule. We have typically these kind of PK profile where we have plasma concentration on the y-axis and time. An administration of a drug will render an increase in exposure as you have an absorption process. If you had given it either IV or subcutaneously, you would perhaps have started somewhere up here. But for the hypothesis of a fictive oral drug, we see that as the drug is absorbed, we have an increase in exposure over time until we reach a Cmax, which is the maximum concentration that we have within a dosing interval. And then when the absorption process is less than the elimination process will take over, and we have a decline in exposure. So the absorption is cancelled out, elimination is the driving force, and the concentrations are declining. Unless we give another dose when we reach down here, the concentration will continue, or the exposure will continue towards zero. The slope of this curve is what defines the half-life of the drug, the PK half-life. We're going to come back to the concept of half-life, and I named this talk Time Matters, and that is because it's really about the different ways of viewing half-life and how you can bring the concept of half-life into designing your experiments. The fraction of the dose that reaches systemic circulation after the liver is called bioavailability. So that is also something that we derive from this relationship. Half-life is a parameter or a value that assesses the duration of pharmacological action through exposure. So it gives you a measure of how long the exposure will remain in the body. Drug elimination is usually a first order process, and it is constant in proportion, or it's a constant proportion of drug lost per time unit. In the example that we have to the left here, it is adapted from a book called Oligo-Based Therapies, Methods and Protocols. And we see that we have this biphasic shape of the curve. So we have early on a rapid decline, and then a longer terminal half-life. And we call the first one the distribution phase, followed by the longer elimination phase. And in this case, it's really from the 28 days onward in this example that we have what we call the elimination phase. And we can divide them into initial versus terminal half-life, depending on which part of the distribution process you're in. The half-life is derived from an equation where we take log 2 divided by the elimination rate constant K. And we can also replace K with either volume of distribution over clearance, which is the same parameters. And these two are two PK parameters that you can derive from your PK curves. We're going to come back to how we look at this. So half-life is dependent on both your volume of distribution and the clearance of the drug. And this is true for all drugs, be it small molecules, large molecules, or oligonucleotides. Half-life has a direct impact on the accumulation of drug in the body. Here we have a graphics showing concentration over time, where we give repeated dosing. So we have concentration here, or exposure, on the y-axis, and we have time on the x-axis. And the arrows here, they note when we give different doses. And the simulated curve here will show you the exposure of the drug over time. It takes always, this is a concept, or it's as close to true as one can get, that it takes 3 to 5 half-lives to reach steady state. So if you have a half-life of 1 day, for example, 24 hours, it will take you between 3 to 5 days to reach your steady state level. And steady state is when you keep dosing and you do not get an incremental higher exposure when you keep dosing. So in this case, we see that after the first, second, third, fourth, fifth and sixth dose, there is no more incremental increase in exposure between these two doses. We have reached a steady state relationship. Also, one parameter to keep in mind here is the C trough, the trough concentration. this is something that we as PK experts often talk to our colleagues that we would like to have a trough concentration, and that is because the trough concentration is directly proportional to the elimination. So therefore it's a very informative time point to take. When designing experiments and clinical studies, it is a very handy concentration or measurement to have, because you can take it right before you dose in the morning. So if it is a preclinical in vivo study, or if it is in a clinical study, you can capture this very easily before you administer the drug at the either in the animal house or at your clinical site, before you dose the next dose. So it's both easy to capture, and it is a very informative time measure, and we're going to come back to that a little bit more in detail. Comparing then, putting this half-life concept into looking at the short versus the long half-life. I've again put in a simulation here where we're comparing a long half-life in blue versus a short half-life in red. And with a short half-life, you quickly reach a steady state level, which means that really after the second dose or third dose, you are more or less at the steady state. While if you have a longer half-life, it will take you a longer time to reach your steady state level. A long half-life may enable you to dose less frequently, because you have, of course, the drug resident for a longer time within the body, and the accumulation is more pronounced. One thing to keep in mind is that with different oligo sequences and different target and targets organs, you may actually have different half-life. So for a small molecule, you might be looking at your plasma concentration half-life, while for oligos, it might be your liver, it might be your kidney, it might be a muscle that is your target organ, and you might have different half-lives in different organs. So therefore, it's good to keep track of which organ you are in and define it where it is most relevant for you. So I said that the C trough value was important, and now I'm going to show you how it's easy to use in order to get a derivation of half-life. So this is a accumulation index curve, an equation that we have put in here. So it's, we have up here, and then we can rearrange this equation into actually getting the half-life out of it. And looking at the actual information that is needed here, it is tau, which is the dosing interval. Log 2 is very easy, that's about 0.7. And then we have this A, those are the unknowns in this one. And A is defined as the accumulation index. So if you know how much your drug is accumulating over time, and you know how often it has been dosed, you can actually derive the half-life for it. And this is a very handy and very easy way to derive the half-life. You do not need to take full profiles of exposure of your compound. You can just monitor these trough values as they increase towards steady state. So looking to the graph, looking at the graph to the right, we can take a C trough value going from about 15 to about 30. It's a bit less here. But if we say that we have an accumulation of a doubling of C trough between day 7 and day 28, the accumulation index is about 2. So it's an then we know the information that we are missing in this equation here. Tau is known because you know how often you dose. In this case, it's dosed every 7 days. So you can put in 7 days here replacing tau, and then putting in 2 as your accumulation index, and by that you get a half-life. And in this case, if you take log 2, which is 0.7, you multiply it by 7, that's about 5, and then divide it by the log of 0.5, that's about minus 0.7, and you come out with about 7 days as your half-life. And you can play around with this, and you can try different and see how accumulation will actually, or how half-life will impact accumulation by altering these terms in the equation. And this equation is also again available in this book that we are referencing here. I see that I have a question coming in on the chat. We have here an example also around how conjugation can impact your profile of your PK profile. The oligo exposure at the target is more relevant, of course, than the plasma concentration. And in this case, we are looking at plasma exposure for a non-conjugated and a conjugated oligo. The disposition of the oligo is sequence and target dependent. So it means that you can have different profiles for different sequences and different target organs. If you reach a saturation in liver uptake, for example, this can lead to a higher plasma concentration. We commonly use mRNA knockdown as a surrogate for exposure in tissue. Those are usually quite well correlated. Sometimes you need to distinguish between productive and non-productive uptake into cells and tissues. And what is that then? You can have uptake into a certain cell type up until a certain level where an increase in exposure will not give you any more effect. And then you have a non-productive uptake. You have a higher uptake into the cell that will not give you any more added effect. We also suggest that quantitating the oligo across tissues of interest for both safety and efficacy purposes is a good way to get a sense of how your oligo is distributed. It can help you with your safety assessment, and it can also give you information around whether you have multiple organs contributing to your disease, or if it is only one cell type or one organ. If you are hitting different cell types, it might be good to do fractionation studies looking at separate cell types, and also if you have different tissues. As we see in this example here with a GalNAc versus a non-GalNAc, we see that they are more efficiently cleared from the plasma if you conjugate them. They go directly to the liver, and therefore the plasma exposure is smaller. Different kind of conjugation can shift your liver-kidney ratios, meaning that if you, for example, conjugate with a GalNAc, you can get a preferential distribution towards a certain organ. With a GalNAc, it is, of course, the hepatocytes and the liver.
We have a question.
I see that I've gotten two questions on the chat, and the first one is around if we can separate productive versus non-productive uptake, and yes, you can. If you run dose response studies where you look at different levels of dosing, you will see that you can reach kind of saturation or maximum if you have a concentration. If you measure that in front before measuring dose, and you see that you have an increase in exposure in your tissue, but you do not reach a higher effect, then you can draw the conclusion that you still have an uptake into that tissue which do not render an effect. So it's a non-productive uptakes. Bioanalysis, yeah, there are different methods you're making. You're suggesting hybridization assay and LCMS. I have seen both. I'm not an expert in bioanalysis, but in my experience, I have seen both, and dependent on the level of sensitivity that you need in your assay, either method can be used with success. So from an Astra perspective, I would say that we maybe use the hybridization assays early on, and then we like to move to more LCMS based further down the line. Oligo, yes, I have a question here as well, if it refers to SI or ASO or both, and I have purposely not divided them into SI and ASO, and I use oligonucleotides interchangeably. There are differences, of course, between them in the metabolism and being single versus double-stranded. But for the purpose of this exercise, I simplify it into oligonucleotides. Thank you very much for your questions, and please send them through as they arise. Okay, so when you have information around the PK of your oligo, you can construct a PK model where you have a dose. It's a mathematical model where you correlate the given dose to some kind of subcutaneous or intravenous or oral compartment that will give rise to a central volume, and then it's distributed both to a peripheral or a deep peripheral volume, and then cleared from the body. When you measure in several different organs, here is an example where you have where we have kidney, liver, and plasma data. You can, of course, add on different tissues. It does not only need to be your plasma data that informs your model building with the purpose of being able to describe, if I give a certain dose, what exposure will it give rise to in plasma, in kidney, and in liver. We will not go into much detail on that other than mentioning that that is a way to predict and make model simulations, and that you use your observed data to inform it. Some general PK parameters and definition of, we have touched upon clearance, which is a measurement of the elimination of a drug. We have the volume of distribution, which is a measure of how the drug is distributed in the body. We talked about the half-life previously, which we derive from clearance and volume. So it's log 2 times the volume of distribution divided by the clearance. Cmax, we talked about, that is the maximum observed concentration or exposure. Tmax is the time to reach that maximum concentration. The trough level is the trough concentration it's the nadir between the two dosing intervals, be it in human or being in an animal study. And it's very easily captured in a study, as you can take that sample right before you give the next dose to your animal or your patient or your healthy volunteer in the morning. So therefore it will give you information on the accumulation of your drug, which you can use to derive your half-life, and it is a convenient one to take from a study operational perspective. We talk about AUC as well, area under the curve, as representative of total drug exposure over time. AUC is classically defined as the bioavailability times the dose divided by the clearance, and it is depicted here to the right as the blue area. It can be divided into two parts. One is, of course, to the last observation. That is, sometimes you see it as AUC 0 to tau, and then you sometimes see AUC 0 to infinity, and that is when you have extrapolated your also to when it is 0. We have another question coming in here. Can the difference in oligonucleotide clearance between GalNAc and non-GalNAc attributed to GalNAc only, or does the stem chemistry play a role as well? That's a very good question. To me, that kind of really comes into what you define as the stem chemistry in this case, what kind of backbone you have, and if that is how stable that is. The benefit of or the when putting on a GalNAc, you reach a metabolizing organ like the liver in a higher extent, and therefore you can impact clearance by conjugating the oligo. Does the antisense oligonucleotide is the same as shRNAs? I will come back to that question a little bit later. We keep moving on right now. So we talked about the other part of the PKPD, which is PD, and we learned from the first half of the presentation that pharmacokinetics is what the drug does, what the body does to the drug, and then pharmacodynamics is then defined as the opposite. It is what the study of what the drug does to the body. So it is really the process at which your drug is impacting your body systems. Classically, pharmacodynamics, you have the Emax Michaelis-Menten curve, where you have an effect set off against the dose and a concentration. You have the Emax function that is down here, where you have either set it out as a dose and you define your ED50, or you have it towards concentration or exposure and you get your EC50. This model or this description of effect does not take into account any time delays. So it is a direct effect model, which means that the concentration that you have or the dose that you have will actually give rise to something that occurs immediately, and that there is no time delay between you putting on your dose or reaching a certain concentration before the effect kicks in. And the time component can be very important when assessing dose response relationship, and we will come back to that a little bit later. This also requires that you have something continuous to measure. So it's a more or less of a biomarker mRNA knockdown, for example, that you can relate to different drug levels. And this is really important to remember here, that PK half-life and effect half-life are not the same thing. So you can have one time resolution for your PK and a completely different one for your PD. And we're going to touch on that in a little bit. For the same parameter definitions that we had for PK, we have similar ones for PD. This is again the same curve where we have mRNA over dose, and we define the baseline as the start-off point, the E0. This you can get either from running a vehicle control or perhaps preferably using a scrambled sequence that will give you the kind of the no effect limit downwards. As you go up in dose, here in this example, you reach the 50% knockdown where you have your ED50. There is a slope parameter which we call gamma for this curve. It can be very, very steep, meaning that if you are in the steep part of the curve, a small increment in dose will give you a big effect change, or it can be a flat curve, meaning that really a quite big change or going up in a big dose increase will give you a quite small incremental effect gain. And that parameter is also estimated when we do these kind of curve estimations. And then your maximum, it can be 0 or if you have some kind of limit, biological limit in your system that you cannot go below a certain value, that will be your maximum effect that you can reach. One thing to think about is that when we put percentages as a percentage towards the baseline, it's a relative inhibition, while if you are looking in absolute values, then you have absolute inhibition. So this is not then expressed in percentages, but rather in actual the same unit as you have your effect in. And good to keep remember that a low value for the slope factor will give you a flat relationship, and a high value will give you a steep relationship until you more or less reach the on-off step where you go from 0 to full effect just by moving a little bit on your dose or exposure curve, or on the x-axis here. So the time component is very important also for effect. We need to remember that when we set off dose or exposure versus either mRNA or protein knockdown, it's a snapshot in time. So it is at one point. While in reality, when you run your study, you have the more or less the same accumulation of effect, dependent on, of course, that you accumulate exposure in your mRNA knockdown. So when you look at mRNA or protein knockdown over time, you have the same as for the PK profile, that within a dosing interval you have an incremental reduction and a fluctuation in the effect. And you will have an accumulation of effect over time, depending on the drug exposure or the effect half-life. So when we talked about the PK profile or the PK half-life, that was the accumulation of your exposure. Likewise, we have the half-life of your effect will determine the accumulation of your effects. And why is this important to know and understand that there is a difference between the snapshot and the time resolution? That is, of course, because that will govern when to measure in your study. And here we are comparing an example. If you measure your knockdown, your PD effect at week 1, comparing it to if you measure at week 5, you will have a big difference in the effect that you actually observe. And therefore, when you do the dose response assessment, there is no concentration that you can tie it up. There is no time adjustment. You will just get what knockdown you have right now with this dose. And if you take that measurement at the wrong time, you might draw the conclusion that it looks less potent than it actually really is, because you have not reached the steady state level that dose response assessment is based on. We talked about the PD half-life, that is the time to reach 50% of the effect, and the onset offset, that is also how quick or how quickly your effect kicks in, and also for how long it stays. The link between exposure and effect over time, you can use something called an indirect response model, where you will have this is your basic PK model. You have a dose giving rise to a concentration in your organ and an elimination from that organ. That concentration or exposure have an inhibitory effect either on the formation or stimulating on the removal of a biomarker, or which give rise to an effect. And in this case, you really account for the time resolution, the time that it takes from the dosing, and it correlates the exposure to the effect and not just the dose response, which is a snapshot in time. So in this example here, where we have a indirect response model, it takes into account both the time course of the oligo exposure and the PD effect. Yes, and of course the impact of making your drug response assessment at the wrong time point is that you will have an underestimate of the effect. If you take it here, of course it's the extremes here. If you take it at week 1, it may look like your drug maybe only reaches a 20-30% reduction, while if you go to the full length, go out to the steady state time point, you actually see that the same repeated dose will give you about 60% effect. So it's a big difference, threefold difference, and the risk that you run is that with taking your assessment at the wrong time is that you actually believe that it is less potent, or you have a less of an effect than you actually have. So when developing drugs and when we take it another step, you also need to keep in mind that the mRNA knockdown that you are looking to get with your oligo or your protein knockdown, it also needs to translate into some kind of pharmacological efficacy. I've put in an example of a disease that I've come across. It's called NASH, non-alcoholic steatohepatitis. It's a accumulation of fat in the liver, which is a side effect of diabetes or other welfare diseases. And in the severe case, it causes cirrhotic liver, and you will need a liver transplant. So in this case, you will need to define kind of where is the knockdown of my target, where does the knockdown of my target need to be in order to render a pharmacologically relevant effect. And typically you can offset it like this, where you have different, it can be fibrosis staging. So if you have different level of fibrosis and or improvements on your y-axis, and you offset that against an mRNA or a protein knockdown, and you define this curve. And also, where is the clinically relevant effect that I want to achieve? Is it 50% effect? Is it 70% effect? And then you translate that to the level of mRNA knockdown that you need to reach. Of course, when designing these kind of pharmacological preclinical studies, but also clinical studies, there is a difference between establishing your proof of concept, meaning that you actually only need to prove that your drug is working in the intended indication, and building a quantitative link. Proving that your drug actually works is of course perfect, because then you have built confidence in it, and you have built reasons to believe, and you have you can really move from that point onwards with confidence. while in order to make predictions and moving outside of the observed realm, you will need to establish a quantitative link between these, and for that you will need, of course, more than one dose to define these kind of curves. You cannot define the shape of this curve with only one dose, because that can be anywhere on this curve and you don't really know. And of course, defining what is the clinically relevant effect in the preclinical setting, that can be very difficult, often something that is informed by literature data. And as you move along into the clinic, it might be when you compare versus a competitor, or what the regulators have defined as that's a minimum effect you need to achieve. So 2 questions has come in. Will mRNA suffice, or is there a need to demonstrate protein knockdown? I would say that depends on how interlinked they are. If you need to have protein as well, and if you, of course, in a perfect world, you would establish exposure mRNA protein hard endpoint, and that link. While you need to, if you need to skip certain steps, perhaps going directly from exposure knockdown into efficacy, if you can build that link without having the protein data, then you can do that. That is my experience, at least. Plasma correlation between Cmax, AUC, and PD effect. For small molecules, there can be a direct link. It depends on where your target is. If it is a liver target, then you will not see the same kind of correlation between plasma exposure and effect. While if it is a circulating target, then perhaps plasma exposure will be very representative of your PD effects that you are seeing. So it depends on where your target is located. Yes, so in conclusion, we will now move on a little bit into pitfalls and study designs. Achieving sufficient in vivo exposure at the target with the route of administration and the oligo half-life and potency will have an impact. They all are interlinked, and the time course is important. The plasma protein binding will determine the distribution and excretion of the oligonucleotide and depend on the sequence. We have touched upon that time is of the essence. You have half-life, both a PK half-life and your PD half-life. Mean they can be either tightly connected, dependent on each other, or they can be separate. And effect biomarkers such as mRNA can be quantitatively related to either dose or exposure, and we typically do that in a multi-dose study, so you have more than one dose. Let's see, go to the next slide. No. So this is a biomarker map. It was first developed by Danhoff et al. And it is available in this publication. This is something that you can for yourself build and try to fill out the gaps. And this is a tool for you to use when developing a new drug or how you could think about driving it forward to a product or a clinical product in patients. So the suggestion is here to define an animal and a human track going from the genotype to the dose to some kind of biomarkers to an endpoint. And we're going to touch upon the different stages and how you can use different study and models in order to build knowledge around this in your animal track, and then do translation between different stages between human and animal in order to build your human dose and also build confidence in your oligo as you go forward. Yes, biomarkers and endpoints. These are defined in this case as biomarkers are continuous parameters. We can have mRNA, but it can also be measuring the leakage of proteins in the urine. It can be LDL levels, it can be HbA1cs, but typically for oligos we have the benefit of being able to measure mRNA. We can use it for differentiation between different oligos, different sequences, and it can be to benchmark against competitors. It can also help us in dose setting, defining what is a safe and a therapeutic level. So it's something that we measure relatively easy. While an endpoint, that's more of your yes and no answer. It can be survival or it can be frequency of events. It's often very predictive of a disease outcome. So of course, if you prolong survival in an animal model, it's most likely it's more predictive than if you just look at the mRNA levels. In the clinical setting, we can use them for registration and claiming benefit, meaning that we use it in the label and also when we submit for a new drug application. And they are, both for safety and efficacy. So the way that you can think about this is to start with kind of defining the disease or what is it do I need to achieve and what data is out there. Here is an example then for non-alcoholic steatohepatitis. Very fortunately, there is a draft guidance from the FDA where it really tells you exactly what you need to achieve in order to develop a new drug for NASH. So it's a resolution of steatohepatitis on overall histopathology readings or no worsening or fibrosis. It is improvement of fibrosis, or it is both resolution of steatohepatitis and improvement of fibrosis. So here in this case it has been so fortunate that someone has defined what you actually need to achieve, and then you can start in your biomarker map to work backwards to really defining and filling out the gaps in the biomarker map. Why don't you always choose a good biomarker instead of an outcome? Why do you need outcome measures? Well, of course, they can be predictive or non-predictive, and sometimes biomarkers like mRNA, they take a much shorter time to develop effect. Here is an example of albuminuria, so it's the leakage of albumin in urine. It's adapted from a publication where they have a small molecule and they measured proteinuria and kidney function, and we see that we have the maximum effect is actually reached after 3 months of treatment. While if you want to have something that is really predictive of disease outcome, then you will need to go to kidney function. But seeing an improvement in kidney function, this is the difference here is that the positive effect is above placebo. Placebo is the dashed line, and here the positive effect is below placebo. You will actually need to dose for more than a year in order to see a positive effect if you have this kind of more endpoint driven biomarker. So therefore we use these perhaps less predictive biomarkers sometimes as a way to get a quick assessment for dose setting and for differentiation and for comparing different oligos, different sequences between each other. While when we really need to claim something, then going into the more endpoint driven studies is recommended. Yes, and also when looking at where to start, genetic association versus acquired phenotype, what kind of human target validation data is available, is there a causality or is there only a consequence, and also thinking about the patient population, what is practical and what is ethical to do. And we use the biomarker map to go backwards and end up with a dose translation and what we actually in the end define that we need to bring into man. let me just go back. And as you see here, there are a couple of studies example here. We have pharmacology studies. So these are your typical mice studies that you would have where you look at mRNA or protein knockdown. It may be correlated to some kind of biomarker. It can be liver triglycerides or whatever, and also that you look at some kind of hard endpoints such as NASH resolution. And that is what you pick up in your regular pharmacology study. Then you might have a transgenic study where you look at the dose to mRNA relationship for your clinical candidate in a transgenic setting. It might also give you the liver oligo concentration versus your mRNA knockdown, and then you might refine it with some kind of primate data if you have that in hand. And then you can do your scaling that, okay, there is a good correlation between achieving your liver oligo exposure to knockdown. That will more probably be the same, and then your mRNA to liver triglycerides might also be similar in human and animals. And then you can do a dose scaling based on a scaling principle that we will come to a little bit later. In the interest of time, I will actually skip this one and move into a discussion around multiple doses and what to think about here. So we want to establish a quantitative relationship between the level of knockdown and the disease modifying effect. And we look upon it as mRNA versus dose, and you might want to go in with 3 doses. But then you also need to keep in mind that in an efficacy study, that mRNA knockdown needs to be correlated to a efficacy versus knockdown. So it's 2 different ways of seeing the effects that you will get in your study. And try to distribute these doses along the curve so that you capture a high knockdown around your ED50 or EC50, and also a lower dose so that you get the shape of the curve. So anchoring your study design around maximum around your ED50 and also a lower one will give you a lot of information. But optimizing it for mRNA or efficacy, that is the efficacy relationship, that is a very different question. And it's rarely that you actually know this relationship quantitatively so that you can make predictions of what mRNA actually will lead to in efficacy. And therefore, this one is much harder to do than this one. And when you interpret your results, of course, from the study, you need to define what the clinically relevant effect level is. Do you need to have a very high level of knockdown that renders a high efficacy, or can you live with a lower level of knockdown? And you also keep in mind that this is a snapshot in time. This is at one time point, and it will depend on when you measure. And your mRNA knockdown has a completely different time resolution than your efficacy. And we're going to show that example really in a little while. These slides here are in here just for repetition. I will skip through them now, but if you go through the presentation later on, they will help you just to repeat what we've talked about now. So here we have an example where we plot mRNA versus protein over time. This is just a simulation of the single dose. And we see that mRNA is usually quite quick to come. We have a quick onset of effect, and it also disappears quite quickly. While the protein, it will take some more time to develop, and it also stays for a longer time. The slope of this curve here gives you the information about the effect half-life. So if you want to know about the half-life for the protein, for example, then it's really good to measure your protein knockdown as this curve returns towards 0. And the same for the mRNA. The slope of this return to baseline following a single dose will will give you information about the time course of knockdown. And the way one would design that in a study is perhaps to give a single dose and then sacrifice animal groups over time as you go back towards zero, if you cannot measure a circulating biomarker that is translatable to your mRNA knockdown. If you want to do it in a multi-dose fashion, or a single dose is not sufficient to get to study the effects that you want to have, then perhaps a multi-dose loading dose scenario might be the best for you. That is when you give doses with a couple of doses with a tight time interval to get a strong effect. And of course, then if you have a hard endpoint that or an effect that you measure in your study, it might have a completely different time resolution than your mRNA and your protein. And if you remember that we talked about that the exposure to mRNA and the mRNA to efficacy, both of them are a snapshot in time. When do you measure when you have this kind of scenario? So it comes back to the question of the linking mRNA to protein to effect. When do you measure? I mean, if you would measure in this case when you have the maximum efficacy, perhaps you will not have any mRNA knockdown because you have it has already disappeared, because it has a quick onset but also a quick offset, while protein, which has a slower onset, has also an even slower return to baseline, and efficacy might be the extreme than in the other case. It has a slow onset but it stays on for a longer time. So really sampling during a recovery phase will give you information of the time course, and given that you have different time resolution on mRNA, on protein and effect, you might actually make false conclusions if you are not aware of when you measure your effect, be it mRNA, protein, or any other hard effect that you look at. So it's very important to keep the time resolution in mind when you design your experiments. Have I measured long enough? Is the study long enough to capture what I want to capture? Have I maximized the effect when I decide to measure? Yeah, and loading doses is of course a way to get quickly up to steady state, which is the level where you will not get any more incremental benefit, be it on mRNA, be it on protein, or be it on efficacy. So if you go back here, now we have defined kind of the relationship between mRNA and up to efficacy. When we think about scaling and interspecies scaling, I'm going to touch upon that a little bit quickly. We are closing to the hour. I don't want to take up much more of the time, but I still want to give you this perspective. So, of course, we cannot use the mouse data that we generated in the pharmacology studies for assessment of potency of our human candidates unless your target is very well preserved between different species. So one way of doing it might be to go through a transgenic mouse strain and look at the potency of your human candidate, and then again define your dose to knockdown relationship. You need to keep in mind that if this is also a snapshot in time, so keeping in mind when the time resolution and when you are at steady state with the human candidate will impact this assessment of potency in also your transgenic mice, if that is the method that you choose. The more doses that you can put in, of course, there is a maximum, but going from 3 dose levels up to maybe 5 dose levels will increase your precision in the estimate of your ED50, and that will be directly proportional to the human dose that you will estimate or that you will predict. So a suggestion, if you want to have a high precision in ED50 or EC50 is actually to go in with more dose levels, perhaps smaller groups. You're not looking for statistical pairwise comparison here. You want to build a quantitative relationship, and you may not need as big groups, but you need to have them more well distributed along the dose response curve in order to really capture the shape, the maximum, and the ED50 value. There is a publication from Anderson et al. That shows an approach on how you can do interspecies scaling, and that for liver targets there is a quite nice correlation between mouse target engagement and human target engagement, and you can actually use this to predict and translate the mouse efficacy, or sorry, the mouse potency data to human, and by that you derive your human dose prediction, and you translate your target engagement in the mouse study to a predicted target engagement in humans. Of course, the caveat is that going to emerging therapies in other tissues than liver, there is less information, there are less clinical data where you have been able to do this translation and build these kind of curves. So therefore the uncertainty is bigger in those kind of targets, but for liver it is quite well defined. We talked about the non-human, the understanding of exposure in different tissues. If your target organ is liver, you might want to refine your PK predictions in a tolerability screen. It can be performed in non-human primate, which is then more predictive of the PK in human, and it is a way to refine PK predictions where you have organ or plasma exposure that you can monitor over time, and then you translate via something called allometric scaling, which we show here, how you correlate the PK parameters between different species. So of course you can, if you want, mouse we know is not very good, but from rat and dog and monkey you can do a body weight based scaling of PK parameters, and by that you get the exposure, the predicted exposure in man, which is then can be related to the potency that we saw scaling of previously in order to derive the human dose. So starting out with a disease, going backwards to your pharmacological models that you have. You can have a transgenic mice in order to build the potency of your human candidate. You might be able to have non-human primate data to make it more robust on your dose to, for example, liver exposure in this case, and then use allometric principles to scale your dose prediction to man. So it's a quite easy structured way in order to set up your different experiments in order to reach what your human dose prediction will be. And of course, this is something that you then will evaluate in your clinical studies, and it will be a justification for the documentation that you used for your first time in man studies. In conclusion, I mean, oligos are thoroughly screened to ensure safety and efficacy. The biomarker map will help you in developing your oligonucleotide drug, and to structure what biomarkers and endpoints to keep track of them, and also what is the clinically relevant effect that you need to achieve. We have looked at the time component, which is very important both for your efficacy, your potency, and your exposure studies, and it may impact your assessment if you take if you make your assessment at the wrong time point. So if you keep track of the time resolution of what you're actually going to measure, it will be much easier for you to end up in a pitfall where you actually think that something is less effective than it really is. One thing to keep in mind is appropriate controls, is that you have maybe both vehicles and scrambled oligos to really being able to distinguish what is a class effect and what is a non-class effect. Just comparing something towards a PBS, you might have pure oligo effects that you otherwise would not capture. And increasing the number of dose groups in dose response studies may increase the precision in your potency assessment. If you're not planning your study for a pairwise comparison, you can shrink your group sizes for a quantitative analysis. And then we have touched upon the scaling principles that can be applied for PK to derive the predicted dose in man. And by that, I would like to say thank you for inviting me and for giving me this talk. And also thank you to those of you who have posed questions. There are a couple of ones that are still open, and we can try address them as we go, if we have a few more minutes. I would also like to acknowledge my colleagues at AstraZeneca, especially Rasmus Jansson Löfmark, Shalini Andersson, Marie Elebring, and Anders Dahlén. So thank you very much.
Thank you very much, Carl. That was a very, very good presentation on this. Yeah, shall we go and quickly go through some questions? I think we had one from Lena. Does the AON the same as shRNA? Is a software that predicts the half-life of our AON target binding?
Yeah. I don't, if antisense oligonucleotides are the same as for saRNA, that I don't know, but we can, I'm not really sure what you mean in that question, and we can take that offline, perhaps. Is there a software that predicts the half-life of our AON target binding? No. There is no black box software that will predict it for you, to my knowledge. You will need to derive it from your assays, and the way to do it is to give perhaps a single dose and then have recovery groups where you look at the time resolution over time. So it doesn't need to be very long studies unless you have a very long half-life, because then you would need to actually design studies that are very long to capture the return to baseline. Yeah. Good. Should we let me? What scaling should be used for oligos between animals and humans, and what is effect of GalNAc? So yeah, that was the slide that I showed. Let me see this one. So if you go into this presentation, I don't think that there is a some of them are conjugated and some are not. So and I don't think that we make any as long as you are at steady state, it shouldn't matter if you have a conjugate or a non-conjugate, because you will have more or less maxed out the effect anyway. But I can recommend this this paper here for how we do the interspecies scaling on effect. For PK, then allometric principles is kind of what we use. If you want to make this line yourself, recommend to have more than 2 species, of course, in order to make an appropriate scaling. That would be my recommendation. Is there anything else?
I'm not sure, did we address Nuno's question on does sequence affect PK properties more than chemical class modifications on oligonucleotides?
Yeah, it's a good question as well. In my experience, where I have seen variations in sequence in terms of exposure. But it can also, since when we screen different sequences, we usually only have kind of mRNA data, so then you can have differences based on that, and it's not always that you have exposure data to make a direct comparison. Where we do see big differences can be in different targets. That's where I would say that you have that really impacts your what half-life you have, depending on which target you're going for.
Okay, are there any more questions? In the meantime, I have a question for you, Carl. I've been told that there is a particular software that people use to do all these calculations. Can you recommend something, or?
I don't want to make any commercials for any specific software. No, I would say that there are, of course, commercially available softwares to derive PK profiles. An example is Phoenix Vinonlin, that I would say is commonly used. There are in R, of course, different types of methods that you can use to derive PK and do PK estimation and simulation. When I work with, when you have bigger data sets, you might want to use MATLAB, you might want to use even more advanced modeling softwares like Monolix and NonMem. But for the examples that I've shown today, you can really make it with more or less GraphPad or very simple software, and usually Excel will get you quite a long way. The important thing is to keep in mind the time resolution, that you have differences, and if you want to estimate parameters, then you will need to have some kind of estimation software in order to do that. And there are more advanced and less advanced ones available.
Thank you very much. I don't think we have any more questions, so I think we should close it here. Just a quick reminder that we will have a webinar on academia or industry, a career discussion on the 15th of July with Professor Annemieke Aerts-Maroes, who is the current OTS president and from Leiden University Medical Center, and Dr. Shalini Anderson, the chief scientist and of new therapeutic modalities and head of oligonucleotide discovery at AstraZeneca. And there's also an OTS Journal Club next week on June the 18th at 14 UTC, and you can sign up for that. I'll post a link into the website. And then thank you very much again, Carl. That was really a very good presentation. One question: will you make the slides available? I think you hadn't decided last week.
Yes, I think we can make them. Just let me check with my legal team once again that I inform them of that, and I will get back to you. But most likely they will. It will not be a problem.
Okay, thank you very much.
Thank you.
And yeah, have a good day, everyone. Bye.
Bye.